Hello viewers, in my last lecture I presented to you an account of some of the pioneering investigations which led to our understanding of the structure of the nucleotides of DNA and RNA. However, nucleic acids were thought to be simple in structure and proteins were still being considered as the main candidates for carrying genetic information. The interest in DNA acids, nucleic acids and DNA was rekindled when three scientists, Avery MacLeod and McCarthy, gave a convincing biochemical proof that DNA is the transforming principle and may function as the genetic material. An Austrian biochemist, Erwin Chargaff, extended Levine's investigations and provided some very valuable data on the structure of DNA. He concluded in 1950 that nucleotide composition of DNA varies among species. According to him, the DNA base composition varies between species, but determines that within a species, the bases in DNA are always present in fixed ratios. That is, the same number of adenines as thymines and same number of cytosines as guanines. In other words, in any one species, the four bases are found in characteristic but not necessarily equal ratios. He also found a peculiar regularity in the ratios of nucleotide bases which are known as a Chargaff's base pair rules. According to Chargaff's rules, and states that in any species there is an equal number of adenines and thymine bases and equal number of guanine and cytosine bases, as I've just said before. So on your screens now that you can see is that number of adenines is equal to number of thymines and number of guanines is equal to number of cytosines. That means the total number of purines is equal to pyridines. After purifying these bases from different species, he separated them by simple technique of paper chromatography. So in other words, we can say that the Chargaff rule states that total number of purines in a DNA molecule is equal to the total number of pyrimidines, that is, A is equal to T and G is equal to C. So, A plus G is equal to T plus C. Thus, the ratio of A plus G to T plus C is equal to 1. But it is not necessary that A plus T is always equal to G plus C. It may be equal, it may not be equal. So, the ratio of A plus T to G plus C is constant and is different for different species. For example, AT-GC ratio for Bacillus bacterium is 0.4, while that of human beings is 1.56. Lesser the AT-GC ratio, more thermostable would be DNA because of higher GC triple bonds as compared to the AT double bonds. And you know that GC triple bonds are stable than AT double bonds. On your screens, I have shared with you the data which shows the composition of AT and GC. And what we find in those species where the percentage of GC is more as compared to A plus T, those species are more thermostable. That is, for example, the DNA of E. coli is more stable than the DNA of humans, because here in case of E. coli the percentage of GC is greater than the percentage of A plus T. The fourth decade of 20th century witnessed a renewed interest to investigate the structure of DNA molecule. Various research groups began X-ray diffraction analysis to study the spatial relationships of different components of the nucleotides. Four major groups of researchers were William T. Astbury, Linus Pauling, Wilkins and Rosalind Franklin, J.D. Watson and F.H.C. Crick. An English physicist and molecular biologist, William Thomson Astbury, made pioneering X-ray diffraction studies of biological molecules. His work on keratin provided the foundation for Linus Pauling's subsequent discovery of the alpha helix. He also studied the structure for DNA in 1937 and made the first step in the elucidation of its structure. Astbury had recognized the fact that DNA produced a diffraction pattern which indicated that it also had a regular structure and it might be feasible to deduce it. Bell and Astbury published an X-ray study on DNA in 1938, describing the nucleotides as a pile of pennies. Astbury and Bell reported that DNA's structure repeated every 2.7 nanometers and that the bases lay flat, stacked 0.34 nanometers apart. Astbury pointed out that the 0.34 nanometer spacing was the same as amino acids in polypeptide chains. In 1946, Astbury presented a paper at a symposium in Cambridge in which he said, and I quote, "Biosynthesis is supremely a question of fitting molecules or parts of molecules against another. And one of the great biological developments of our time is the realization that probably the most fundamental interaction of all is that between proteins and nucleic acids." He also said that the spacing between the nucleotides and the spacing of amino acids in proteins was not an arithmetical accident. The biological significance of this still remains unexplained. Another Doyen, an American chemist, biochemist, chemical engineer, and a peace activist, Linus Pauling, made very significant contributions to our understanding of nucleic acids and DNA. He won a Nobel Prize in Chemistry in 1954 for his research into the nature of the chemical bond and its application to the elucidation of the structure of complex substances. He also got a Nobel Peace Prize in 1962. That means Linus Pauling received two prizes, that is, Nobel Prize in Chemistry in 1954 and Nobel Prize, Peace Prize in 1962. While working on proteins, he became interested in DNA. And early in 1953, he, along with another crystallographer, R. Corey, published their version of DNA structure. That is, 3 strands twisted around each other in rope-like fashion. According to Pauling, and I quote, "We have formulated a structure for the nucleic acids. The structure involves 3 intertwined helical polynucleotide chains. Each chain has approximately 24 nucleotide residues in 7 turns of the helix. The helixes form a right-handed screw. The phosphate groups are closely packed about the axis, with the pentose residue and the purines and pyrimidines groups projecting readily." I unquote. So he had laid the foundation that the helix of DNA was a right-handed helix. So on your screens now you can see the structure which the Pauling had introduced, and according to him, the phosphate groups were in the center around the axis, and the sugars and the bases were projecting readily outwards. This model was later proved to be wrong. Nevertheless, Pauling's investigations raised several questions that stimulated the future workers to investigate the DNA structure further. Another group, led by New Zealand-born British biophysicist M.H.F. Wilkins, was active at King's College, London. He started work on the X-ray diffraction analysis of DNA molecules. He was later joined by Rosalind Franklin. So, Maurice Wilkins and Rosalind Franklin worked together with X-ray studies. And Rosalind Franklin was a British chemist and an ace X-ray crystallographer whose work was central to the understanding of the molecular structure of DNA, RNA, viruses, coal, and graphite. On your screens, you can see the instrument with which she studied the X-ray analysis of the DNA molecule. Franklin is best known for her work on the X-ray diffraction images of DNA while at King's College, London. Particularly, photo 51 taken by her student Raymond Gosling. This photo 51 was acquired through 100 hours of X-ray exposure from a machine Franklin herself had refined. From this pattern, she deduced the basic dimensions of DNA strands. She showed that DNA had a regularly repeating helical structure and the phosphates were on the outside of the helix. On your screens now you can see the historical picture of DNA, in which Franklin showed that it had a double helix structure, which is on the right hand side. She also showed the distances between the two bases and also showed the distance, the one complete round of the helix, that one complete turn of the helix. And on the picture on the left, she showed the tilt of the helix. So this is a very historical picture which laid the foundation for Watson and Crick's double helix model. Maurice Wilkins and Max Peruz, who later himself got the Nobel Prize in 1962 for his X-ray work on blood, shared this photo number 51 with James D. Watson, who was working along with Crick. They learnt from this data the helical shape, which helped them to deduce the width of the helix and the space between the bases. Unfortunately, Franklin died of ovarian cancer in 1958 at the young age of 37. Franklin's images allowed James Watson and Francis Crick to create their famous two-strand or double helix model, for which Francis Crick, James Watson and Maurice Wilkins shared the Nobel Prize in Physiology or Medicines in 1962. Franklin's contribution to the discovery of the structure of DNA were largely unrecognized during her life, for which she has been variously referred to as the wronged heroine, the dark lady of the DNA, the forgotten heroine, a feminist icon of molecular biology. James Watson and Francis Crick were working at Cambridge, where they began to construct three-dimensional molecular model of DNA. Both of them were brilliant chemists and had tremendous insights. They tested their hunches on the basis of other people's work. Their concept building and logic to formulate the double helix model of DNA was based on several lines of earlier experimental evidences. First, the molecular modeling of Pauling. Second, on X-ray diffraction studies by Franklin. Third, on the basis of Bayes' rules by Erwin Chargaff. Their genius, insight, and imagination combined all these concepts. Their modeling efforts were also motivated by several questions raised by Pauling as to how many strands are there in a DNA molecule? Is the number of strands constant for every DNA helix? Are the bases pointed towards the helical axis or away from it? What are the explicit angles and coordinates of all the bonds and atoms? Watson and Crick built physical models using metals, rods and balls, in which they incorporated the known chemical structures of the nucleotides as well as the known position of the linkages joining one nucleotide to the next along the polymer. On your screens, you can see the original wire and ball model of the DNA double helix as proposed by Watson and Crick. Watson and Crick began to work on DNA strand with two strands, the double helix. Using molecular models of wire, they first tried to place the sugar phosphate chains on the inside. However, this did not fit with the X-ray measurements and other information on the chemistry of DNA. The key breakthrough came when Watson put the sugar phosphate chain on the outside and the nitrogen bases on the inside of the double helix. The sugar phosphate chains of each strand are like the side ropes of a rope ladder. Pairs of nitrogen bases, one from each strand, forms rungs, the bars that form the steps of a ladder. The ladder forms a twist every 10 bases. The nitrogenous bases are paired in specific combinations: adenine with thymine and guanine with cytosine. However, the pairing of like nucleotides did not fit the uniform diameter indicated by X-ray data. A purine-purine pair would be too wide. A pyrimidine-pyrimidine pair will be too short. So, only a pyrimidine-purine pairing would produce the 2 nanometer diameter indicated by the X-ray data. So, on your screens now you can see that the size of purines is large. So, if one purine makes a bond with the second purine, the distance would be too wide, whereas the size of pyrimidines is small. So, if a pyrimidine makes a bond with pyrimidine, the distance would be too narrow. So, this distance would be exact if the purine makes a bond with pyrimidine, and this data, which is very accurate, was consistent with the X-ray data. So, it was presumed, it was said that a purine is making a bond with a pyrimidine. In addition, Watson and Crick determined that chemical side groups of nitrogen bases would form hydrogen bonds, which will connect the two strands. According to them, adenine would form 2 hydrogen bonds only with thymine. So, the linkage is going to be between adenine and thymine. And guanine would form 3 hydrogen bonds only with cytosine. Therefore, the linkage has to be between guanine and cytosine. These findings were consistent with the Chargaff's rules. That is, the base pairing rules also justify the combinations of nitrogenous bases that form the rungs of DNA. However, the sequence of nucleotides along each DNA strand is not restricted. Any of the 80 GC pairs can be aligned. Thus, the linear sequence of 4 bases can be different in countless ways. But each gene has a unique order of nitrogenous bases. On your screens, you can see the first sketch of DNA double helix as drawn by Watson and Crick. And in this, you can see that the side ropes would be containing the sugars and the phosphate, and the rings which you can see are the rungs where the bases are present. So now on your screens you can see the double helix structure as proposed by Watson and Crick, where the side chains that is the ropes are representing the sugars and the phosphate residues, whereas the rungs, the bonds joining these two chains, are representing the bases. That is, adenine is linked to thymine and cytosine is linked to uracil. In April 1953, Watson and Crick published a 1-page paper in Nature on the structure of DNA. Double helix has been hailed as the greatest discovery of the 20th century. James Watson, in his book The Double Helix, which was published in 1968, said, "A structure," and I quote, "a structure this pretty just had to exist." For this lecture, I had referred to the books by Brucker, Watson, Peirce, Klug and co-workers. So when Watson said that a structure so pretty had to exist, we shall be discussing the structure of this beautiful molecule in great details in our coming lecture. Thank you.